Welcome to a recent study on the multimodal analysis of ultra-short to long-chain PFAS by LCMS. Work was performed by Christine Amador, PhD candidate at the Colorado School of Mines, Alexander Shrum, U.S. Product Manager at Showa America in New York City, and Hiroki Takanaka at Showa Japan. In our presentation, we'll briefly discuss the history of PFAS, the separation modes, including ion pairing, and ion exchange helic as a multi-mode alternative, instrumentation used in our method development, the method development itself, results and discussion, and some concluding remarks. Perfluoroalkyls and polyfluoroalkyls, or PFAS, are a subset of a family of fluorinated chemicals. These chemicals were first developed in the 1930s, but came into widespread use in the late 1960s, especially after a fiery naval accident in 1967. Today, these compounds are used in the manufacture of products including non-stick, heat-resistant surface coatings, firefighting and non-flammable foams, food packaging linings, and adhesives. In the graphics, you can see additional products that PFAS are found in, including pesticides, cleaning products, paints and sealants, and even dental floss. PFAS comprise an incredibly wide variety of over 3,000 different compounds. In the words of Ian Cousins and colleagues from the 2020 article, Strategies for Grouping Per and Polyfluoroalkyl Substances, PFAS, to Protect Human and Environmental Health. The PFAS class comprises distinct substances with very different structures and properties: high molecular weight polymers and high molecular weight nonpolymers, neutral anionic, cationic, and zwitterionic substances, solids, liquids, and gases, highly reactive and non-reactive inert substances, soluble and insoluble substances, and volatile and involatile substances. In the environment, some PFAS are mobile and others are immobile, and some bioaccumulate while others do not. Please refer to the references at the end of this presentation for a link to this article. The Interstate Technology Regulatory Council, or ITRC, defines two primary classes, namely polymers and nonpolymers. Polymer PFAS may contain a CH backbone with fluorinated side chains or other complex forms, whereas nonpolymers comprise either a molecule where each carbon is bonded with only fluorine atoms, or may contain at least one carbon-hydrogen bond within the backbone structure. Nomenclature is defined as all CF bonds plus the carboxylate carbon or sulfonate group at the end of the chain. For example, perfluorooctanoic acid contains 7 carbons bonded to fluorine plus 1 carbon in the carboxylate moiety, or 8 carbons total, hence octanoic. In the schematic, polymers are shown on the right. Polymers like PTFE and PFPE are of low concern due to no or very low bioaccumulation. However, they are still closely studied. Moreover, polymer PFAS can be precursors to non-polymer compounds. Current monitoring and remediation is concentrated on non-polymer compounds. The CF bond is one of the strongest covalent bonds in chemistry. Thus, PFAS are not prone to dissociation and release of the fluorine atom under normal environmental conditions. Further, the carboxylate or sulfonate end groups allow for bioabsorption, leading to many possible negative health outcomes. PFAS are stable under environmental conditions, and many PFAS, especially C1 to C10 chain length compounds, remain in the soil and can quickly migrate into groundwater and eventually to the drinking water supply. Bioaccumulation is associated with many increased health risks, including thyroid disease, testicular cancer, and lower birth weight. Today, it is a primary concern to detect and remove PFAS from soils and water supplies. Constant monitoring is essential. There are several ways to monitor soils and water supplies. High-performance liquid chromatography is often employed for all water monitoring, including groundwater, drinking water, and wastewater. Moreover, liquid chromatography mass spectrometry, or LC-MS, is used for more sensitive analysis as local and federal governments continue to reduce the acceptable limits of contamination, now in the low parts per trillion, or PPT, range. Traditionally, reversed phase chromatography, or RP-HPLC, was used for the separation of hydrocarbon polymer chains. Reverse phase works well as the hydrophobic chains are easily partitioned between the C10 solid support functional groups and the eluting hydrocarbon chains. However, partitioning becomes more complex in PFAS with hydrophilic terminal groups like carboxylates and sulfonates. This becomes worse with shorter chains. The compound becomes more ionic, resisting interaction with the C10 groups. To restore effective partitioning, an ion pair reagent can be employed to interact with both the analyte and the solid support, as shown in the schematic. Ion pair reagents have several drawbacks, however. They can't be used in LC-MS analysis. Well, they can, but it creates problems in the future. We'll get back to this one in a minute. Ion pair reagents are temperature sensitive, and elution is highly temperature dependent and must be carefully controlled. Ion pair reagents require slow equilibration time, thus large volumes of eluent are required. Due to the long equilibration time, gradient elution is not possible. Most ion pair reagents highly absorb in the UV range, thus making UV detection difficult. Most importantly, most reagents cannot be completely removed from the column or the system. That means even if they are successfully used in mass spec analysis, they remain in the system, contributing to high backgrounds and problems in future LC-MS analysis. Multimodal or mixed mode analysis utilizes a solid support with strong anion exchange functional groups. An ionic buffer and an organic, such as acetonitrile, is added to the mobile phase to regulate the elution of the analytes. Multimodal analysis is proposed as an alternative to ion pairing for a few reasons. It works well, especially for ultra-short, short, and long chain analysis. This includes the C1 to C10 compounds. Equilibration time is short. Gradient elution can be used. The use of mass spectrometric friendly salts like ammonium bicarbonate can be used to easily control elution and peak resolution. 3 instruments were used for method development, including a Shimadzu I-Series 2050 equipped with a UV detector for initial development at Shodex in New York City, a Waters Acuity UPLC H-Class SQD2 single quad ion trap in Japan, and an Agilent 1200 coupled to a SciEx 5500 Q trap at the Colorado School of Mines for final LC-MS calibration curve and sample analyses. The chosen HPLC column was the Shodex VT-50 2D. The column dimensions were 2.0 by 150 millimeters in peak housing. The housing was packed with a solid support made of polyvinyl alcohol particles with quaternary ammonium functional groups. Samples were dissolved in 50% methanol for best volatility and ionization. Sulfonate PFAS species sensitivity was consistent with both buffers. However, sensitivity of carboxylic acid PFAS species improved significantly by using an acetate buffer as shown in the figures. Therefore, ammonium acetate buffer was selected for final development. The ratio of buffer to acetonitrile was adjusted to observe the highest resolution versus the lowest retention time. Moreover, final studies included observations of the strongest signal-to-noise ratios for each compound. Buffer concentration and pH have the largest effect on retention time and peak resolution. The key may be the very high hydrophobicity of the CF backbone as compared to a hydrocarbon backbone. At high pH, pH 8.6 in this case, there are less free protons in solution. PFAS are more soluble in acetonitrile and less likely to retain on the solid support versus the organic buffer, acetate for example. Thus we see a reduction in retention time. Long-chain PFAS are very hydrophobic. They are more affected by the organic solvent versus the hydrophilic terminal groups and less affected by higher pH. Reducing the buffer concentration to 25 millimolar in this case has a similar effect for long-chain PFAS. A reduction in the buffer reduces the ionic interactions between PFAS and buffer. Again, the organic solvent, acetonitrile, increases the solubility and the compounds elute faster. However, short-chain PFAS hydrophobicity is lower. Short-chain PFAS compete and retain on the solid support due to the ammonium pore mobile phase and are less solubilized by the organic. Performance was best using 26% ammonium acetate buffer and 74% acetonitrile at a pH of 6.8 and a flow rate of 0.2 mils per minute. The oven temperature was set at 40 degrees. You can see all the instrument and sample parameters on the screen here. The optimized ammonium acetate method was able to simultaneously retain most ultra-short through long-chain compounds and showed linear calibration curves for TFA, PFPRA, and PFBS through long-chain compounds. Representative calibration curves are shown on the screen. These are cal curves for TFMS, TFA, TriFBA, and PFOA. Sensitivity was good down to 1 part per billion. The PFAS were separated and eluted in the order of larger to smaller chain length, showing multi-modal retention characteristics, including anion exchange, with the highest signal-to-noise ratios with the use of 70 to 80% acetonitrile. The method was unable to detect DFA on all instruments, but was detected during method development by LCMS. Although MFA was not detected under the current method conditions, it is possible that further optimization and ionization may allow detection of the remaining ultra-short compounds to be detected. Chloroacetic acid species had similar retention characteristics to corresponding organofluoride acetic acids during method development. In conclusion, the Shodex Helix-Pack VT-50 2D with a PVA solid support and quaternary ammonium functional groups demonstrated highly selective LC-ESI-MS measurements. The simple isocratic mixture of 50 millimolar aqueous ammonium acetate solution and acetonitrile enabled the simultaneous analysis of ultra-short through long-chain PFAS, which cannot be simultaneously retained using reverse phase ion pair reagent methods. Further method development includes better understanding of the mixed mode effect on elution, mass spec ionization parameter optimization, lower detection limits, especially for ultra short chain compounds, and cross-instrument validation. Please see the references on the concluding slide. A final, more in-depth presentation will be available in early to mid-August. You will also have access to our poster on this topic beginning on June 28th. Thank you.